Okay, so today we're going to talk about the UA SRC DMAC Olympus web server and its integration with the Cyverse. Okay, and we are rolling out beta testing phase. So we have been in development phase for a while, and now finally we are getting ready for beta rollout. So after today colloquium, hopefully people will sign up and start using it and do bug reporting for us so we can fix it and make it better. So the Olympus web application server, we name it Olympus as in the Mount Olympus that is home to all the Greek god and goddesses. And it homes to web applications that name after the Greek god and goddesses. Right now we have the low hanging fruit, I call it low hanging fruit because these are apps that are easier to develop. We have Ares, Demeter, and Hermes, and there are many more to come. And what Ares, Demeter, and Hermes are, they are essentially part of a RNA-seq data analysis pipeline that will bring you from raw FASTQ files. So if you send your sample to UAGC to do RNA sequencing, we have worked with the UAGC, so they'll directly deposit your data into your folder in Cyverse UA SRC drive. So your data file will be ready in Cyverse for you, and after that you have the FASTQ file, and from there you can use Demeter, Ares, and Hermes, bring your data all the way to predictive pathway analysis, right? And what these are is essentially these are low-hanging fruit. What we have created is a graphical user interface. You have user-friendly graphical user interface wrapper around popular command-based software like Star Aligner. There are popular command-based R scripts like DE6, and also Cluster Profiler. So Ares is essentially running DE6. Demeter is essentially running Star Aligner, and also Bowtie, which I've forgotten to put over here, but Michelle just reminded me earlier. And Hermes is actually running Cluster Profiler, which is itself is a wrapper for GSEA and overlap analysis developed by Broad Institute. And how this thing works is like this. So you perform your experiment, you submit your sample for, let's say, RNA-seq by the UAGC core. UAGC core will upload those data onto Cyverse for you on your account, and then you can activate Demeter. You can input, you can customize it by enter your own parameter in Demeter, Ares, and Hermes. Ultimately, you're going to get predictive pathway and tell you what to look for in your data, right? And another thing that why we are doing this is also for this type of analysis, usually lab that carry out a lot of this analysis themselves. Like my lab, we have a dedicated workstation that do this kind of stuff, right? So I have a computer that costs $12,000 that I use to do this kind of stuff because it uses a lot of CPU and memory. But right now, Demeter is actually doing all the heavy lifting for you in Cyverse using computing resources available to us in Cyverse. So essentially, Cyverse has a lot of supercomputer and computing power, and Cyverse will carry out authentication for us. So only UA SRC member will have access to these resources. So you will have to sign up for an account with Cyverse, and then you can log in, and authentication is done on Cyverse. Demeter is essentially like a portal to interact with Cyverse and using Cyverse computing resources to carry out the computation for us. And I would like to think of it something like this, we're building like a DMAC virtual machine. You have Cyverse out there, and then this is Mount Olympus, home to all the great god and goddesses. And we are forming a virtual link between them. And this machine actually do all the analysis for us. And in time, we'll add more and more analysis, and researcher can suggest to us what kind of apps that they want us to develop, and we'll add to it as well. So without further ado, today we are going to do live demo. I'm a little bit nervous about this, but Michelle, who is the creator for Demeter, is going to show us how to actually upload data to Cyverse. Let's say you have old FASTQ file that you want to analyze, you could also do that, but you have to upload it to Cyverse, because once your files is in Cyverse, uh, Cyverse will do all the tracking for you. So, we make sure our data follow the FAIR criteria. We can track it right from the beginning to the end, right? So Cyverse to do the tracking for you. Michelle will show you how to sign that up and how to use Demeter. And then the creator, Aries and Hermes, Anthony, will take over and explain to you how to do that. And at the end, Michelle will also talk to you about how to upload data to Cyverse, and also how to sign up, put your name in a Google form that she created. All right, so Michelle, take over.
All right. Hello, everybody. Okay. All right, so just to follow up on what Ike said, everything that we're showing today is specific to RNA sequencing data, but as you follow along, just be thinking about, if you're not working with RNA sequencing data, think about your own data and what sort of tools you think would be useful for us to build for your data. We're definitely excited to talk to all of you about your data and your workflows to build tools that will be useful for you. So with that, I'm going to first talk a little bit about how to use Cyverse, and then I'll jump into the demo of some of our apps. This is the Cyverse discovery environment that I'm showing you. The discovery environment is the primary web interface that you'll use to interact with your data on Cyverse. So you can access the discovery environment at sonora.cyverse.org. All you have to do is create an account, and then you're able to upload data to Cyverse. So this is my homepage. And to go to my data, I'll just go over to this data tab on the left-hand side. And this is my home folder on Cyverse. Every single user gets a home folder. I think you get up to like 10 gigabytes of storage, where you can put whatever data you want.
100 gigs.
Huh?
100.
100? Oh, okay. Wow. Okay. 100 gigs of data. And yeah, this is like your own space. It's private to you, but you can also share data with other Cyverse users, which is really easy to do. For example, if I want to share one of my data folders with some of my colleagues, I can just go over here and hit share. And as long as they have a Cyverse account, I can just type in their username. I can share this folder with Ike with just a few clicks. You can give them read or write access. So one super useful feature of Cyverse, you can just share data very easily. But for our SRP data, we're not going to be storing those in our home folders. We've created a shared folder under the community data section. So our shared folder, it's a little bit down. It's called SRP DMAC, and we're going to give access to all of the Superfund members to this folder. Right now we have it organized by project. So depending on which project you're working under, that's where your data will go. And then additionally, under each project folder, we've created folders for each investigator. So each lab will have their dedicated folder to deposit data into. But for today's demo, we're going to use Ike Sang's lab folder as an example. And since he's not under any of the projects, it's just under this DMAC folder, under the UA folder. And the way that we've organized this is that every file extension or file type has its own folder. So for example, in Ike Sang's lab, all of the FASTQ folders or all of the FASTQ files will go under the FASTQ folder, and you can either upload your own or have them put there by UAGC. And then like all the BAM files will go into the BAM folder, and all the SAM files go into the SAM folder, and so on. But for today, we're going to be running some analysis on FASTQ files. So we'll go ahead and go into that folder. And Ixing's uploaded a bunch of FASTQ files that need to be analyzed. So to do that, we're going to head over to our apps at our DMAC website. So our DMAC website is at dmac.pharmacy.arizona.edu. And so we've put everything related to DMAC here. We have a contact us page with our email address. We've also set up a discussion forum. We're hoping this, as we all start to use Cyverse, we can use this as an area to all help each other. As well, we can also talk about like new app ideas or ideas for data pipelines to integrate into Cyverse and stuff like that. We've also set up a YouTube channel where we'll put any videos that we create here. So Nirav gave a talk to the Superfund trainees a little while ago. So we've uploaded that here. Today's talk will be uploaded here as well. And we'll be creating additional video resources about like how to upload data to Cyverse and stuff like that. But if we go back to our homepage, this is also where we can access all the apps that we've developed. So right now there's only 3, but there will be many more in the future. So the first step in our data pipeline, like Ixing was talking about, is Demeter, where we're going to be mapping the FASTQ files to their reference genomes using the Star Aligner software. So we'll go ahead and launch that. And then in the Demeter home, basically what you'll see is an exact mirror of all the FASTQ files you have uploaded to Cyverse. So if we go back to the discovery environment, we can see that these files are the same as the ones that you see in Demeter. And then we can go ahead and launch our Star Aligner analysis. So the DNA sequencing option will do like the bow tie 2 analysis, and then RNA sequencing will run the Star Aligner. So we've built an interface that allows you to easily group your FASTQ files. I'll just run maybe two analyses. And then you can select your reference genome here, as well as the read length. And then when you click submit, these are actually analyses that will be submitted to Cyverse, and they'll be running on Cyverse. So hopefully this will go through. All right, so in the discovery environment, you can see that we've gotten some new notifications that new analyses have started running. So those are the 2 I just submitted. And if we want to look at a completed analysis, I ran this earlier today. You can go to the output folder. And so these are all the files that were generated as part of the analysis we just ran. For example, we can click on one of the files and look at the results of the mapping. And then this reads per gene file actually automatically is getting transferred to our DMAC Olympus server to use with the Aries app with the DESeq2. And so for this part of the demo, I'm going to pass it off to Anthony to show you guys how that works.
All right, thank you, Michelle. So I'm just going to share my screen here. All right, so as you've seen, we're going to go along the pipeline here. So we can launch Aries. There we can see the analysis from Hermes is inputted here. And as we were talking about before, DSeq looks for differentially expressed genes. And so this is an example of experiment where there was an NRF2 knockout. So we have 2 NRF2 knockout replicates here, and then we have 2 wild types without the knockout. So if we want to compare the 2 wild types to the 2 knockouts to see if we can find any differentially expressed genes, we can just select them and hit submit, and then we can see it's loading up here. So it usually takes a few minutes to do, and we get an output here. And we can see we have the group 1 or the wild types that we selected, and group 2 are the knockouts. We get this nice volcano plot, which is interactive. So we can hover over specific points, and we can look at things like the gene specific gene name, the base mean, the log2 fold change, the adjusted p-value. We have a line here of the p-value greater than 0.05, so everything above this line is significant. We have some pretty cool features here to explore this. We can filter out specific parts. So if we want to look at only the genes that are above 5 with their low fold change, we can just type in 5 here. And we'll see that it slowly starts taking away everything else. Sometimes it can be a little slow, but it gets the job done. Let's say we want to do some other types of filtering here. We can say if we want to look at only log 10 adjusted values over, let's say 150, we can get rid of these down here. Just type in 150. And then here we have a subset here. We can tag points. So we can highlight and select specific points. And we can get, right now it's selecting the fold change values, but we can go down and say select gene name. And here we have a bunch of the gene names associated with the genes in this area. So cool things about this that we can do, we can save this. It's an SVG. And then just open it up here. We see it's the exact same we just were looking at. We can clear the tags and highlight different areas and do those sort of things. And there's a nice table at the bottom with some of the data laid out. We can also download this as a CSV. The only thing I would say, I would not recommend highlighting everything because then it tries to tag everything and that takes a while. So my only advice is be careful what you tag. So moving over, if you want to take this analysis here and push it to the next part of our analysis, we can select the name. For this, we're just going to call it H383 and just hit save. So we'll go home, and now that we have our differential gene expression analysis, we can try to do the gene set enrichment analysis and pathway analysis. So we'll just launch this. And here we see that file that we just saved. For this demo, we can select, for Hermes, we have a lot of different databases we can use for disease ontology, and we can explore all these different ones. For now, we're going to do molecular signature database, and specifically, we're going to look at motif gene sets. So when we're doing this, we can choose whether we want to look at downregulated genes or upregulated genes, or we can do some combination of both, or we cut off different sides. For now, we're just going to do the both. So this takes a few minutes to run. It's running pretty intensive. While we wait, I can answer any questions anybody might have up to this point.
So I have a question. The Cyverse, I guess you need to have an account to be able to access. But if these apps are residing in the Superfund website, do these need a Cyverse account to access?
Yes, the Cyverse account is linked with this. So basically you log in to these apps through Cyverse.
But you had the Superfund information is all inside Cyverse.
Correct. So there's a special form we're going to talk about at the end that you have to fill out to give us your Cyverse username and email. So then we link your Cyverse username and email to our apps here, so you'll be able to access them specifically. Okay. If that makes sense.
Following on that question, this is not linked to the Superfund website, is that correct?
No, that is correct. Yeah, so the Superfund website is is more sort of a informational portal. This is a different system altogether.
There is a link that Michelle put on the landing page, which is dmac.pharmacy.arizona.edu.
Uh-huh.
You go there, you click learn more, it will bring you to, there you go. And then you can also click on Superfund. You'll go to our main Superfund website. If you did over there, if you click on Superfund, you'll go to Superfund. I think Michelle has included, I mean, there are links that go there. Yeah.
Right.
There you go.
So you can, what you're saying is you can find the Superfund website from this DMAC site.
Correct.
Can you find this DMAC site from the Superfund website or not?
Not yet, but
We can
that
put
I
it-
think
we can
Lucille
put
can
it
help
up
us do
there.
that.
So, Reena, short answer, once we are ready with,
You will. Okay.
then we will connect. Right now, we are
And-
sort of in the
Yeah, and the reason we do all this login stuff and authentication through Cyverse is that in the future we're planning on open this up to other Superfund program from other university, right? And then we can do data integration between Superfund program.
Right. I think that's going to be really powerful. I was just trying to clarify, not criticizing in any way. I just wanted to understand.
Yeah,
That
as
was
it is,
very exciting.
Cyverse already being used by university and all agency already. Yeah.
So excuse me, so then basically then we just provide the raw data, the RNA-seq data, then all the analysis are done by the core, then we just get the results. Is that correct?
No, you can do it yourself on using this web application.
So okay, so the analysis is still done by ourselves then. So
you
we
can
have
do it yourself. If you don't want to do it yourself, you can get us to do it. But this is for you to do it yourself so you can explore your data. And if you want to do more in-depth analysis, we can help you with that.
And will you be setting up appointments with each project to talk about the needs of each project?
Not yet, but we'll do
No,
that.
I mean in the future.
In the future, yes.
Okay.
Yeah. So Anthony.
All right. If we're all set, then I'll continue on here.
So
we see.
I actually have
Go
one
ahead.
more question. Sorry.
Mhm.
So the data input, I mean, the ones that we've been using outside companies to do RNA-seq analysis, right? Are those data easy to import into Cyverse?
Yes, Michelle will show you how to do that after Anthony.
Okay, all right.
Okay, so once we've launched analysis, we can have a summary here, which files we selected. And then we have, as we see the wild types here versus the knockouts. And we come here, we have the standard GSEA plot. Then we can select which specific motifs we want to look at, and it's organized by p-value. And here we see that here we have a cutoff, so everything above this line is significant. We see the NRF2 is one of the significant terms. We also see that we have some things closely related with this NRF2 here, so we're just going to select some of them. And we can submit analysis, and then we can look at the graphs overlaid. Here, one of the next visualizations we have is a gene concept network. So we can look at the overlap between specific motifs and specific genes. And so we can see like this gene right here overlaps between these three motifs. This is a little cluttered, so if you want to take a less cluttered, we can just sort it only by category. Hit submit. And so then while that's running, we'll move on to the next one. Here we can look at the log2 fold distribution for each one of these motifs. And so we see here NRF2 is log fold is over 0, which makes sense because it's should be upregulated. And some of these related motifs here as well, we see here. And last, we have a dot plot that looks at the gene ratio. And here we see the NRF2 has a very high gene ratio. Yeah, we can kind of go back, see how some of these things. So we can come back here and look at all these overlaid and like this GSEA plot. We can also download this as an SVG and save it for yourself. So this is basically the gene set enrichment analysis, and here's a less cluttered version of that previous graph. And we can sort it by different types of things. If you only want to look at the genes, if you want to look at everything, if you don't want anything at all. And we have the colored by the logful change. So this is this area. We also have overrepresentation enrichment analysis. So about the overrepresentation enrichment analysis, we can actually rerun the whole analysis here based on whether we want to look at downregulated or upregulated genes. And again, this is looking at the overlap in the networks. If we want to, in this case, right, we want to look at upregulated genes. So we can just say we want anything with the log fold change greater than 1. Select only categories. We don't want the cluttered look and submit it. Here we have an enrichment map, which pretty much shows us the overlap between motifs that have similar genes inside of them. And again, I'm just going to rerun this with log fold change greater than 1 to look at upregulated genes. Here we have an upset overlap plot, which basically is a fancy Venn diagram where we can see specific motifs which have overlapping genes and how many overlapping genes they have. So right here we have these 4 or 5 motifs. I have about this many overlapping genes, and we can take a visualization at that. So again, I'm going to submit this with the greater log fold change. And then last, we kind of have the same dot plot we were looking at before. And we can display how many you want to look at. I can look at the top 10, top 20 is the default, top 30. I'll go see the other. So if we come back to here, we see this NRF2 has been highlighted, and then this is closely associated. And we can kind of see that some of these genes over here are overlapping between the two. We can go over here and look at some of these structures and see that the NRF2 gene, again, is overlapping with this TCF11 gene, or motif. So these motifs kind of create the structure here. We can come here again and look at the overlap now that we've rerun it. And we can say, if we want to look at the overlap between the NRF2 and the NCF11, kind of see that it's got a high level of gene overlap here. And then we can go back to the dot plot, and we kind of see NRF2 here and some related genes and the overrepresentation analysis. So here's some of the visualization tools you have available to you. You can kind of play with different parameters, different upregulation, different downregulating. You can play with different p-value cutoffs. So the last thing I want to show here is that we also have this KEGG pathway analysis. But again, it's going to take a few minutes, and I kind of went over a lot there. If we want to stop and talk about anything, we can.
Yes, I have a question. Either Anthony or Exxon can answer it. So what do you mean NRF2 and the TCF have overlapping motif?
So TCF11 is NRF1. They call it TCF11.
Oh my God, no wonder. Okay, so that they have overlap sequence.
Yeah, the recognition sequence. So yeah, so you see the algorithm accurately pick up the NRF2 knockout experiment, right? We knock out NRF2, and then the algorithm go in and pick up, oh, this NRF2 is the main change here.
Yeah, okay.
And then one of the last tools you'll have with this pathway, we get an extra option here of this pathway analysis. And basically you'll get a look at different pathways, and you can see which specific genes along this pathway are upregulated or downregulated. And again, this is organized by p-value. So these are the highest p-value to the lowest p-value to the highest p-value. And here we have our cutoffs. Everything below this is insignificant. So if you want to look at, say, ferroptosis, and then it'll basically run the analysis and under the ferroptosis, and you can look at the pathway to see which genes inside of them, again, are overregulated or downregulated. And got this visual here, and again, you can download all of these different visuals. That's a summary of what Hermes does. And so I think I'll kick it back to Michelle to talk about what sort of, or how to upload your data.
Yeah, so if you want to use these resources, how do you get access to them? Michelle gonna explain that, and how, if you have your own FASTQ file that is not run by UAGC, Michelle can show you how to upload that onto Cyverse and use it.
Can I ask a quick question before Michelle starts? Sorry.
Yeah, of course.
How's this relate to David?
Oh, David performed functional annotation clustering. Is not predictive. So this is gene set enrichment analysis. This is predictive.
Okay. All right. Thank you. Go ahead, Michelle.
Okay. All right. So, hold on. All right. So to upload your files to Cyverse for use with these apps and any future apps that we develop, I'm going to go over 2 ways to upload your data to Cyverse. So let's head back to our SRP DMAC folder. So like we said, like you would deposit your data into your folder for your lab, but we've also created this like sandbox folder if you want to like practice uploading data to Cyverse. So we're going to use that for today. So the first way that you can upload data to Cyverse is through the discovery environment. So let me find my files. You can use this upload button over here, or you can also just drag and drop files from your computer to the web interface. So you can see that this file was queued for upload and that it uploaded already. So that's the first way. The web interface is good for small files. I think maybe like no bigger than 2 gigabytes, depending on your internet connection. It will take a long time to upload. So the second way that you can upload data to Cyverse is using a tool called CyberDuck. And CyberDuck is a free software that you can download online, and we'll provide links to that and everything. There's a little bit of setup involved, but once you set it up, you can connect to Cyverse, and it's pretty easy to upload
And
files.
you can leave that connected throughout the day, right? You can
What?
leave it like your folder on your computer, basically.
Right, so you can, like, if it's a really big file, like you can leave it uploading to CyberDuck just in the background while you're doing other things. Whereas the web interface, like if you close your browser, then the upload will stop. So CyberDuck is a good option. So when I connect to CyberDuck, it first shows my home folder, my Cyverse home folder. If we want to upload data to the SRP folder, we can go to that folder, which is at iPlant/home/shared/SRP_DMAC. And we'll go ahead and go to that folder. And then this is the project structure that I showed earlier. And if we go into the sandbox folder, we can just grab any file from your computer and just drag it into CyberDuck, and it'll start uploading, and you can just leave this going in the background for however long it takes to upload. Yeah, so that's pretty much it for uploading data. We'll be creating like a YouTube video to show like all the details of setting up CyberDuck and stuff, but it's a pretty simple.
Sign up for Cyverse account, right?
Oh yeah, so to be able to upload data, you first have to sign up for a Cyverse account, which is at user.cyverse.org. It's a pretty quick sign up process. And then after you sign up for your account, we've set up this Google form for you to submit your username and the email address that you use to sign up, as well as like what lab you're a part of, the PI project. And I mean, obviously, if you're the PI, like we'll know who you are. But if you have like grad students working under you or any other people working in your lab, they can also sign up where they can work with the data on Cyverse as well.
Michelle?
Yeah.
I think it would be useful to add a couple more folders. So you have projects 1 through 5, but we have a fairly active research translation core that generates data sets.
Yeah, we can definitely add new folders or we can talk to you about which folders you think we should add.
So that's one, and the other one is if Carlotta Chief would like a community engagement folder, and I'm not sure if she does or not, so I'll have to ask her. I think she's here.
Yeah, we can definitely set up other folders as
Okay.
well.
So it would be those two.
Okay.
Yeah, and Raina also, if there are projects that are constantly producing data and they need automation for that data to directly go here, that's also very feasible for us to share. So
Okay.
now that we have this framework, depending on who's doing what, these are Lego blocks that we can connect different things and, you know, make it easy for that group to analyze and share whatever they're doing with this DMAC and beyond this DMAC.
Good.
And then Michelle, we have a 24-hour rule, right? Do you want to explain that?
Oh, well, it's not implemented quite
Okay,
yet.
not yet.
But the idea that we have is that once you upload your data to your Cyverse folder, you have like 24 hours to either like delete it or modify it, just so that we can make sure that our data is not being messed with after it gets uploaded. Yeah, we haven't implemented that yet, but that's our plan for that.
And that's more so to do with things that are like your golden copy, as in something coming off the machine or some level 0. But whatever's in your home directory, you can do whatever you feel like. It's what you give so that other people who are building workflows on your data, they are not taken aback by the fact that you've changed something. So if you bring in data that is to be given to people, once you bring it in, you have 24 hours to change it. But once you contribute it, now it's part of the system. Now, if you have to change that due to some issues, yes, we can. But these are the kind of policies that we collectively, as in all of us, have to decide what will make sure that we are all productive and not just putting data and then realizing and taking it away, and the next person's taking that data and doing the next step to only find that it's no longer there. So it's more to do with reproducibility as well.
I have a question. How about old data from Affymetrix CDNA arrays?
Okay, yeah, that is a good question. So I thought about developing app for those type of data, but I don't know how many people want to use it. So we haven't worked on it yet. We do this because you're low hanging fruit. A lot of people are doing RNA-seq nowadays, and but if there are people who want to use the Affymetrix array, those can be easily done. Yeah.
But I mean, the analysis part should be the same. It's just the data format needs to be changed, right?
No, so Affymetrix array, it uses this robust multi-chip averaging algorithm. So
Yeah.
it's very different from the way you process RNA-seq data. RNA-seq data, like, they're discrete data. So the algorithm is different. So there's going to be, for the processing the raw data is going to be different. But the pathway analysis, you can submit it to Hermes, that will be the same. Yeah, that can be the same. Yeah.
But would be the data format be compatible? I mean, I haven't done this for a long time.
Yeah.
I don't know. Do they have a FastTech this kind of file, or is it something that needs to be converted?
The file is going to be for Affymetrix. It's going to be the cell file.cel file. Yeah, if you have the .cel file, so if we create an app for Affymetrix array, then you just have to upload .cel to Cyverse, and the app will be able to do that. I don't know how many people want that functionality. Is certainly something we can do very easily.
Yeah, we definitely could use that. We have a bunch of data from before that we've done it, but never had time to analyze it, or never had the expertise to help with analyzing.
So okay, so but then if we have the RNA-seq data and we want to the core expert to analyze that, how long it will take to get those meaningful results?
Don't know. Very good question. I do not have an answer for that. We'll have to see what experimental design and and what you want to look at, the scope of the analysis that you want to get done, right?
Okay. All right. We can talk later.
So I guess you're focused right now on sequencing data, right?
Mm-hmm.
But we are going to be generating a lot of geochemical data.
Yes. That is the data I'm interested in. Yes.
Yeah. So I think it would be helpful to the people who are collecting those data sets to hear from you kind of what your timeline is. Are you going to be ready to consider those data sets in a few months or in a half a year, or what kind of is your vision for developing this low-hanging fruit and the apps that you're developing?
Yeah, this is kind of the first phase of it, and Nirav and I have been talking about incorporating what Ramona has created, the annotation, the ontology that Ramona has created into an app that people can use. And also, we are also looking into getting tox scan data. Like NIEHS has a bunch of this toxicology data.
Mm-hmm.
And then we're going to develop apps that based on those databases also. But geochemical data, we'll have to see the data, like see what we can do with the data and what kind of app we can create for those data. And ultimately, all this geospatial data, sequencing data, ontology, everything has to come together, integrate, and we want to create a visualization tool for people to look at how this data kind of integrate together. That's the ultimate goal.
Right. Yeah. And so I'm pushing here a little bit. So what's your timeline? So what should I tell the PIs of these other projects to expect? When should they start interacting with you guys?
Let's try end of the year, third quarter.
Third quarter.
Third quarter.
Yeah. No, that's fine. I just would like to have an idea what to tell people.
I mean, we can start looking at the data now and do some planning. Yeah.
What?
Right now, Anthony and Michelle's are doing all the heavy lifting. We have meeting once a week.
Okay.
So Exxon, those is going to be, as you said, later on will be available for other Superfund people, right?
Yeah.
So, but obviously the data when you're doing analysis is at a very early stage of publication.
Yeah.
So I'm worried about how confidential,
Oh yeah. They cannot access your data unless you share them with them. You share the data with them on Cyverse.
Right. But if you share with them, they publish before you.
Yeah, then don't share with them, right? If you haven't published the data, don't share with them.
So right now, who have access? Our Superfund member?
The tool, only our Superfund member have access to the tool, but data, only you have access to your data.
Oh, I see.
Yeah. So once you uploaded your data onto
so like mine is in group 1 folder, right?
Yeah.
So even group 2, even she cannot see group 1's data?
Yes, unless you share them.
I see.
Yeah.
Okay. All right.
So Michelle showed you earlier, you can share with
I see. Yeah. Okay.
Hey, Donna, I wanted to make sure that I can't see her data. See? That's very important. See?
And Nirav, the Cyverse is the team function that we create team, right?
Yeah, so we can create a team. Yeah, we can then create multiple teams.
Yeah, which can multiple different project can build different project data who's under the same team, right? Basically.
That is correct. So you can create your favorite apps and things and then share it among the team. Yeah. So Donna can decide who is part of it.
Yeah.
And Donna, we can make data look like it's somewhere else so that nobody else can find it but you. So don't worry about it.
So for those who are interested in running, say Demeter, like not using the graphical user interface, you can also launch an application on Cyverse itself. That's another way to run analysis also. If they are interested, Michelle can show how to do that, create webinar or maybe like a YouTube video. So as earlier we showed, we have YouTube account, which you can go and see YouTube video. There's also the forum that you can go and ask questions, and hopefully user can help answer the questions, or one of us can help answer the questions.
So to get access, everybody has to apply a account and the passwords, right?
Oh yeah, it's free. So you just have to sign up for it. Yeah.
So everybody, like in my group, have own different.
Yeah, whoever want to use it, who is in Superfund, they can. Yeah.
Huh.
Yeah, so everyone in your group can create their own Cyverse account, but we'll give all of you access to like your data folder, so you can all work with the data.
So can I ask a question? This is John Shorover. Thanks very much for this presentation. It's been really interesting. I'm curious, who developed the Hermes and Aries apps that you guys showed us? Were those developed by you guys, or was it an interface between these existing apps and Cyverse that was developed?
So
does that make sense?
These apps are wrapper for command line software.
Okay.
Okay, and the interface, the graphical user interface that we showed, like Michelle demonstrated and Anthony demonstrated earlier, those interfaces are all created by Michelle and Anthony.
I see. Because I'm trying to think of how, what would the analog be with the kinds of geochemical data, for example, that we're working with, and what way could we best interface with the data management core to take advantage of that synergy that we would have with you guys. I mean, we have models that we use typically for modeling our data. We input data and we can perform calculations with those data to propose, to create images and things of that sort. But they're likely to be certain types of modeling work that we would like to do that may not exist in those existing apps. And so I'm trying to understand how we would work with you guys to develop something that would actually, be significantly beneficial to all of us, that would warrant the kind of work that you would have to put in to working with us on it. And that's not entirely clear to me. And that may be, that may be my problem. I'm having a hard time still seeing exactly how that's going to work. Yeah, sorry.
I think that's why I asked all the questions about the timeline before.
Right.
I think what's going to have to happen is that Michelle and Anthony are going to sit down with each group at some point, and they are going to ask all of these questions that you're asking of you, right? What are your data sets? What do your data sets look like? How big are they? What do you want to do with them, right? Do you want to interface your data sets with some of these RNA-seq or ITAG data sets? So I think this has to be a process that's going to occur over the next half a year, and that will really involve input from each project to the DMAC so that they can be guided by your needs.
Yeah. And John, just to sort of draw a broad outline to it, you may have your programs that are doing specific things. What we tend to do is look at it at multiple levels. At the most basic level, if your model is written in a specific language, needs specific things that are, what we do is put them in a software container so that it can run by itself, and then make it easy for the web interface to sit on top of it. And by virtue of it being able to partition things with the data, so people can bring in their data and put that in Cyverse, connect your analysis to it, and then be able to launch it without you having to worry whether it's a class full of 30 people or it's a single investigator. Now, making it do all of those steps, those are already baked into what Cyverse has, but the look and feel to make it easy for your community who may not understand the model or may not know the parameters for it, you can make that interface be a little bit easier for them, put some interactive aspects of it so it's easy for a person not to also shoot themselves in the foot. And that's the kind of conversation we'll need to have with you and make sure that it's productive for the group that's going to consume it. I don't know if that sort of helps you to think of it in that abstractions.
Yes, it helps very much. Thank you.
If your software are command line based software, I think the integration, for us to develop like a graphical user interface over it, that is very easy because as Dira alluded to, in Cyverse, they have, the Docker container base where we can create Cyverse apps that run on it, and then a graphical user layer that actually interact with Cyverse that allow people the graphic feel, which is essentially how Demeter is, right? Demeter is a C++ software. The Star Aligner is a C++ software that run on command line. And what Michelle did was create a Docker container that run it on Cyverse, and then a graphical user interface that interact with Cyverse to enter parameter that way.
Excellent.
Yeah.
Are you going to also put something to analyzing proteomic, if not phosphoproteomic?
Yes. How many people want to do proteomic? That's another question. If there's a lot of need, we can develop apps for it. Yeah. If there's not a lot of need, then we can always do it more like a customized way. These apps are essentially allow what we are trying to do is we empower the researcher to play with the data themselves so they can explore data themselves. And if they're interested in more detailed analysis, if they find something interesting, they want to explore further, they can contact us. We can do more in-depth analysis for them,
I have a follow-up security question.
Yeah.
If one of my students wants to look at Donna's data and they sign up and say Donna is their PI, can they see Donna's data?
No.
Yeah, well, that is the thing that like Google Form is supposed to take care of that. That's why we want to cross-check with Rocio, right? To make sure that whoever signed up with Google Form is legit.
Okay.
Yeah. But Rena, short answer. If you sign up, you cannot see my data. If I sign up, I cannot see your data till you explicitly give that person permission. So by default, you can only see what's public inside of Cyverse and what is yours.
But if I have 5 people on my project, since I'm the PI, do I control who has access?
Yes. So you can say here are the 5 people, and you can give them varying level of permissions.
Gotcha.
You can even designate one of the person and say, this is my lab manager. So anybody new that comes in, they can add permission, which is completely okay. You don't have to do that.
Gotcha.
So it's not like everything has to be routed through you,
Okay.
but you want to give that highest level of permission to the most trusted person.
Yeah.
Otherwise, if they delete everything, then it's gone. Now
Okay,
there is a trash can, and you can like go dumpster diving and get your data back.
They can delete it. Once we implemented 24-hour rule, they cannot delete anything.
No. So actually, I'm just talking broadly inside of Cyverse.
Yeah.
So people
I just was curious how the security worked, and it sounds really good.
Yeah, it's very finely controlled, and the part that Akseng is talking about is even one level higher, where the data has a policy that if it's coming from a machine or from some trusted source into DMAC after 24 hours, it cannot be modified. And if you want to, then you have to ask us to help you out. But our hope is that a policy like that will prevent some of the challenges people have. And one additional thing I want to sort of, today was more about DMAC, but Michelle also showed you the discovery environment, and that's where you can go and use other things. For example, you want RStudio to do single cell, or you want Jupyter Notebooks, or you have an Shiny app, doesn't matter. All of those things are available there. I see Powell, so if you want to do Chime, there's a whole version of Chime that's already ready to go. People use that for all sorts of things. So that's already available for you.
You mentioned single cell. Is that also in the pipeline?
Yeah, not in this DMAC pipeline, but there is a single cell. So Surat and other applications are there. So Paul, if you want, I'm happy to show you where they are available.
Yeah, at some point right now, I have a license to Partech Flow. I don't know if you're familiar with it, which is very similar to what you guys are doing, except that they are charging a ton of money for that. But it's essentially a web interface to all of the common tools in RNA-seq and single cell RNA-seq. You may want to look into it, actually. You may have some good ideas how to further develop this. They actually did a really, really thorough and a good job. It's a very intuitive platform. The company is called Partech.
I think I've heard that they used to do a lot of accelerated computing a while back. So the name sounds familiar, but maybe we can, when we come visit your team, Powell, for things, maybe I'll set up some time with you to come take a look. And I'll also bring Ritu, who's in Cyverse. She does a lot of single cell work with different groups. And so maybe it'd be worthwhile. So if there are things that you need and we have, and it's not in Partech, you can use it there as well.
Right. No, I mean, I would be happy to migrate away from Partech, because it would save me, probably $12,000 a year in licensing fees. It would be fantastic, especially that they are using really tools that are otherwise free if you know how to use them in our environment, for example. They're just integrating everything in a very intuitive flow. That's why they even call this suite Partech Flow. I'll be happy to show it to you on my computer.
Is it doing the stochastic one of those principal component analysis we hired to mention?
Oh yeah.
Yeah.
It's a very, very comprehensive platform, and all of the newest tools that come out for single cell genomics usually make its way to this platform within a year or two. So I would highly recommend that you look into it. I know that looking into it means acquiring a license, and it's a pricey endeavor. So I'll be happy to.
It'll be great to see, because we usually work with authors of packages, and so there are so many open source package authors that come to us and say, hey, I want to make this available to the community. And our process is such that if you are even teaching a class full of people, like Chime, where you need decent amount of memory, and so there are Jupyter notebooks that are created, and people just do their analysis using it. So even the Chime people will say, Greg's lab will say, oh, you want to do this, go into Cyverse and run this there. So it would be awesome to see what you are doing and see if there's a way to share that with others, like through.
Yeah. So for the microbiome, we use less Chime, more data too. Right.
Yeah, we have data too. We have that also.
Michelle can make a video on how to launch apps on Cyverse. I think that would be useful. Yeah.
I think we should absolutely do that. I think Paul will. I'll send you an email, and we'll do a Zoom session with you if you can give us maybe half an hour of your time.
Yeah, certainly.
Okay.
But I think a lot of people are going into the single cell genomics, single cell RNA-seq field, and it would be really awesome to have a toolbox for these people that would be intuitive enough, that would not necessarily require a deep knowledge of bioinformatics to use. And I think that is the goal of what you guys are doing, and I can't stress enough how appreciated that is.
And I think I've learned that through Accent, because I'm more of I'm going to teach you how to use complicated tools, and Accent's like, no, people need a simple tool. We have to build this on top of your complicated tools. So it's a yin and yang between me and Accent.
All right, any other questions, comments? So
we're definitely happy to look into the applications that John talked about. We should definitely meet up and discuss like what kind of app we can develop for John and also for you, Reina.
Yep.
Like, the geospatial and also something that more environmental focused apps.
So
that is in our phase 2. And Nirav and I were discussing about creating the whole Indian mythology based apps. Now we're Greek mythology, right? So
I told him there are too many Indian gods, so we won't have the problem of losing names. So
well, so you let us know when you're ready, and my group would be real interested in meeting with you, as would John's group, and probably Mark as well.
Yeah.
So thank you to Xeng and Nirav and Michelle and Anthony. This is great, and probably we should do this again in 3 or 4 or 5 months, right?
Oh yes, we'll have a lot more to show.
Right, exactly. And so I appreciate it, and I think it's really helpful for the center to hear this.
Yeah. And I think, Reina, now that we have the foundation laid out, we'll also do some smaller focused activities and then prioritize what DMAC needs to provide to the broader Superfund at the University of Arizona. So I think that's a collective decision as well, so that the team is delivering value to the whole project.
Yeah, I agree. All right, so everybody keep well, and we'll see you all in a month again.
Thank you.
Bye.
Bye.
Thank you, Anthony and Michelle. Bye, everybody.
Bye.